Imagine two rodents in a laboratory. They look, well, absolutely identical. I mean, same fur, same size, same environment.
But completely indistinguishable to the naked eye.
Exactly.
Right.
But behaviorally, they belong to entirely different universes. One is a fiercely loyal, monogamous partner. It mates for life. The male actively co-parents the pups, and they show this profound distress when separated.
And the other is pretty much a promiscuous loner. It mates, leaves immediately, and shows absolutely zero interest in raising its young.
Right. And the difference between a lifetime of devotion and total isolation isn't a heart or a soul or, a different upbringing. It is a microscopic shift in the map of a single neuropeptide receptor in the brain.
It's honestly one of the most humbling discoveries in modern neuroscience.
Mm.
When we look at the prairie vole versus the montane vole, we're forced to confront the reality that our most profound, complex social behaviors, things that we consider the bedrock of human character, like loyalty and attachment, are fundamentally governed by the physical distribution of neurochemicals.
It's staggering, and today we are opening up the mechanics behind those neurochemicals. We're doing a deep dive into Chapter 1.8 from Kaplan and Sadok's comprehensive textbook of psychiatry, focusing specifically on neuropeptides.
Which is a brilliant topic, especially for those in clinical training.
Definitely. And to conceptualize this for you, the listener, I want you to imagine the human brain as a grand piano. The classical neurotransmitters you learn about in Biology 101, glutamate, GABA, dopamine, these are the pianist's fingers striking the keys.
Right, they're the fast, immediate, moment-to-moment electrical signals. A key is struck, a note rings out. It's clean, binary, and incredibly rapid.
Exactly. Fast neurotransmission acts on the order of milliseconds, right?
Yeah, it provides the immediate data transfer required to, say, move a muscle, or blink an eye, or register a sharp pain.
But a piano piece isn't just about the isolated keys being struck. It's about the resonance, the sustain, the emotional weight of the chords lingering in the air long after the pianist's finger has lifted.
I love that analogy.
If the classical neurotransmitters are the fingers on the keys, neuropeptides are the piano's pedals. They don't just play a single note. They sustain the sound. They alter the resonance. They shift the key and change the emotional tone of the entire instrument over a long period.
They really are the slow signals of significance. That framing gets to the absolute core of their function. Neuropeptides don't merely transmit raw sensory or motor data. They actually shape the biological context in which all that data is interpreted.
Right, they set the mood of the room.
Precisely. They operate over much longer timescales, minutes, hours, sometimes even days. And they orchestrate our most enduring states: chronic stress, social bonding, baseline anxiety, and crucially, the lingering echoes of trauma.
So our mission today is to build a robust mental model of these slow signals. Whether you're a psychiatry trainee preparing for boards, a registrar honing your clinical reasoning, or just a motivated learner, we are moving beyond simply listing facts.
Yeah, we need to understand how psychiatry bridges the gap between a biological molecule and a lived human experience.
Exactly. So let's start by defining the physical object itself. What actually distinguishes a neuropeptide from, say, the classical monoamines?
Well, structurally, a neuropeptide is a chain of amino acids linked by peptide bonds. They are relatively short. We see chains ranging from just 2 amino acids like carnosine to chains over 40 amino acids long, such as corticotropin releasing factor, or CRF.
And there's a biological cutoff there, right?
Yes, the conventional cutoff dictates that once a chain exceeds 90 amino acids, it folds into more complex tertiary structures, and then we classify it as a protein. So neuropeptides are essentially miniature proteins.
Got it. Miniature proteins.
But what's fascinating here is their sheer diversity. We have identified over 100 unique biologically active neuropeptides in the central nervous system.
Which is a staggering cast of characters compared to the, handful of classical transmitters that usually dominate psychopharmacology discussions.
Oh, absolutely. It's a massive system.
Let's test this concept practically for the listener. If I'm a trainee trying to distinguish a neuropeptide's function from glutamate on a microscopic level, what is the fundamental difference in how they deliver their message?
The difference really lies in the receptor target and the spatial release. Glutamate crosses a microscopic synaptic cleft and binds directly to a ligand-gated ion channel. It physically pops open a pore in the membrane, sodium rushes in, and the nerve fires.
Right. It's very point-to-point, highly localized communication.
Exactly. Neuropeptides, on the other hand, rarely open ion channels directly. They act as neuromodulators via G-protein coupled receptors, or GPCRs, initiating secondary messenger cascades.
Okay, let's avoid the biochemical alphabet soup for a second. What does a secondary messenger cascade actually look like inside the neuron?
Think of it as a molecular domino effect. The neuropeptide binds to the outside of the receptor. This changes the physical shape of the receptor on the inside of the cell membrane, which knocks over the first domino, a G protein.
Okay, so the G protein is loose inside the cell.
Right? It travels along the inside of the membrane and activates an amplifier enzyme. That enzyme then floods the internal fluid of the cell with thousands of secondary messenger molecules.
Like CAMP or something similar.
Exactly. And these messengers can travel deep into the cell, altering the resting electrical charge, or, most importantly, they can enter the nucleus and literally change which genes the neuron is expressing.
Wow. So they rewrite the software of the cell, which perfectly explains why the psychological effects of neuropeptides, like the enduring anxiety after a panic attack or the prolonged warmth of a social interaction, last so much longer than the initial trigger.
Exactly. And furthermore, they aren't restricted to that tiny synaptic gap. Neuropeptides utilize what we call volume neurotransmission.
Volume neurotransmission. Unpack that for us.
So when released, they diffuse widely through the extracellular fluid, traveling significant distances to reach multiple target cells. They bathe entire neural circuits simultaneously. They can even be released from non-neuronal glial cells.
Okay, let's unpack this. So if classic transmitters are a text message sent to a specific person, neuropeptides are like a broadcasting system changing the mood of the whole room.
That's a perfect analog. They change the baseline tone of the entire environment.
Which paints a picture of a very fluid, diffuse brain environment. But looking at the source material, there is a fascinating and honestly clinically maddening evolutionary quirk about these molecules.
Ah, yes, the dual function paradox.
Right, they lead a double life.
They really do. Throughout evolutionary history, the body is highly conservative. It rarely invents a new molecule if an old one can be repurposed. Consequently, many neuropeptides evolved one highly specific function within the central nervous system and a completely distinct physiological function in the periphery of the body outside the blood-brain barrier.
Give us a classic example of this.
Well, consider oxytocin and vasopressin. Inside the brain, these peptides regulate trust, emotionality, and pair bonding. But peripherally, oxytocin's primary role is to stimulate profound uterine contractions during parturition childbirth and to trigger the milk ejection reflex during lactation.
Same exact molecule, utterly different jobs.
Yes, and vasopressin, meanwhile, acts on the kidneys as an antidiuretic hormone, instructing the renal tubules to retain water, and it acts on blood vessels to induce severe vasoconstriction.
And the pain system utilizes this exact same evolutionary shortcut, doesn't it?
Indeed it does. The endogenous opioids, acting on mu-opioid receptors. In the central nervous system, these regulate nociception, the perception of pain, and they modulate the mesolimbic reward circuitry.
But outside the brain.
Peripherally, those exact same mu-opioid receptors are densely packed into the enteric nervous system of the gut, where they paralyze gut motility. And they're in the brainstem nuclei that regulate the autonomic drive to breathe.
Which perfectly explains the two greatest hurdles in pain medicine. If a clinician prescribes an exogenous opiate like morphine to target the central pain receptors, the drug circulates systemically.
And it inevitably hits the peripheral receptors.
Right, resulting in profound, often intractable constipation, and at higher doses, fatal respiratory depression.
Precisely. This dual-purpose nature makes targeted drug development incredibly tricky. If you design a synthetic molecule to manipulate a neuropeptide system for a psychiatric condition, you're playing Russian roulette with the peripheral organs, unless you can ensure the drug is strictly confined to the brain.
We will definitely get into the blood-brain barrier problem later because it has derailed literally billions of dollars in pharmaceutical research. But first, let's look at the biology of slow signaling. How does the neuron actually build these petals? Because unlike dopamine, which is synthesized right at the nerve terminal, neuropeptides require a massive logistical operation.
It resembles a specialized heavy machinery manufacturing plant. Monoamines are synthesized locally, an enzyme alters a circulating amino acid at the terminal, and it's ready to go. Neuropeptide synthesis, however, must begin at the DNA level within the cell body, or soma.
So right in the nucleus?
Yes. The DNA gene is transcribed into mRNA, which moves to the rough endoplasmic reticulum. But the translation process doesn't produce the tiny, active neuropeptide. It produces a massive, inactive precursor molecule called a preprohormone.
I always picture the pre-pro hormone as this massive, solid billet of raw steel arriving at the factory floor. It has all the necessary material, but it's completely useless in its current shape.
That's a very fitting analogy. The pre-pro hormone refers to a specific signal peptide sequence at the very front of the molecule. This acts as a sort of molecular transport pallet, guiding the massive protein into the inner chambers of the endoplasmic reticulum.
And once it's inside...
Once inside, an enzyme shears off the transport pallet, leaving us with the prohormone.
So the raw steel is off the pallet and on the assembly line. Now it needs to be carved.
Exactly. The prohormone is shuttled into the Golgi apparatus, packaged into large, dense core secretory granules, and begins a long journey down the axon. Inside these traveling granules, the precision carving occurs. Enzymes known as prohormone convertases act as molecular laser cutters.
And their role is highly specific, isn't it? Let's clarify this mechanism for the listener. What exactly are these convertases targeting?
Prohormone convertases scan the long prohormone chain, looking for specific pairs of basic amino acids, usually pairs of lysine and arginine. When they find these specific pairs, they cleave the chain right at that exact location.
So they're looking for a very specific sequence to make the cut.
Right. And this highly regulated chopping process reduces the massive precursor into the smaller biologically active neuropeptides. And what's remarkable is the biological economy of this system. A single prohormone billet can contain the sequences for multiple different active peptides.
Oh, wow. Really?
Yeah. The prohormone for neurotensin, for example, is cleaved to yield not just neurotensin, but a second active peptide called neuromidin N.
I marvel at that efficiency. But the manufacturing isn't always finished there, is it? The textbook notes that if the factory line stops at cleavage, some of these peptides are still completely inert.
That's right. Many require post-translational modifications to function or to survive the harsh extracellular environment. They might undergo amidation at their carboxy terminus, which acts like an armor plating against degrading enzymes.
Okay, what about TRH? I remember that being a weird one.
Yes, thyrotropin-releasing hormone. For TRH, an enzyme must cyclase a glutamine residue at the front end to yield a specific pyroglutamyl moiety. Without that precise final structural twist, the TRH molecule simply cannot bind to its receptor.
Okay, so the neuron has manufactured these heavily modified peptides, packed them into dense core granules, and shipped them to the terminal. How do they actually get released into the brain?
Well, neuropeptide release is fundamentally different from classical neurotransmitters. Classical transmitters sit in small, clear vesicles right against the synaptic membrane, ready to fuse and release with a single standard action potential.
Right, they're primed and ready.
But neuropeptide granules are large, heavy, and they sit further back from the membrane. To mobilize them, the neuron cannot simply fire once. It must engage in high frequency or burst firing.
So the neuron has to be highly agitated. A whisper won't do it. It essentially has to scream.
Correct. Burst firing causes a massive, prolonged influx of intracellular calcium. Only when calcium levels rise significantly higher than the baseline required for fast neurotransmission will those dense core granules mobilize, fuse with the membrane, and dump their neuropeptides.
Which introduces a really critical concept, co-transmission. Because if a neuron is firing hard enough to release its neuropeptides, it must be simultaneously dumping massive amounts of its classical neurotransmitters. They share the same workspace. How do they interact?
This raises an important question about modulation. Neuropeptides are almost always co-localized and co-released with classical transmitters. They serve to modulate the very signal they are released with. A prime example is neuropeptide Y, or NPY, which is co-released with norepinephrine from sympathetic neurons.
Walk us through the clinical logic of that pairing. Norepinephrine is the acute stress fight-or-flight transmitter. Why release a slow peptide alongside it?
Imagine a severe psychosocial stressor. The locus coeruleus fires rapidly, flooding the forebrain with norepinephrine to drive hyperarousal and vigilance. But if that noradrenergic signal continues unabated, it rapidly leads to cellular toxicity, panic, and structural damage.
It's too much of a good thing.
Exactly. So as the firing rate peaks, the neuron begins co-releasing NPY. NPY diffuses back across the synapse and binds to presynaptic autoreceptors on the very cell that just released it.
It acts as a shutoff switch, an emergency brake.
Precisely. It provides critical negative feedback. It inhibits further release of norepinephrine, ensuring the stress response is intense enough to survive the threat, but self-limiting enough to prevent the nervous system from burning itself out.
Okay, so the signal has been sent. The peptide has done its job modifying the network. How is the signal terminated? With dopamine or serotonin, we have presynaptic reuptake pumps that vacuum the transmitter back up. which is the exact mechanism SSRI antidepressants target.
There are no reuptake pumps for neuropeptides. Their termination is entirely destructive.
It's just a demolition.
Pretty much. The extracellular fluid is rich with enzymes called peptidases. Once a neuropeptide diffuses away from the receptor, these peptidases aggressively cleave the peptide bonds, shredding the molecule into inactive fragments and eventually back into raw amino acids.
So it is a one-way trip. Once released, it is destroyed.
Which means neuropeptides have a very short half-life in the active space, typically measured in minutes. Clinically, this offers a theoretical therapeutic window. If we could develop drugs that inhibit specific brain peptidases, we could artificially prolong the life of endogenous neuropeptides, boosting their signal.
Like ACE inhibitors for blood pressure, right?
Yeah. That is exactly how ACE inhibitors work in general medicine. They block the peptidase that degrades angiotensin. But in psychiatry, developing peptidase inhibitors that can cross the blood-brain barrier and target specific behavioral circuits remains an elusive goal.
We've established the mechanics. We have the factory, the release, the receptor dominoes, and the destructive cleanup. Now we must bridge the gap between these microscopic mechanisms and human suffering. Here's where it gets really interesting. Let's map these peptides onto clinical psychopathology. We're going to look at how specific psychiatric conditions are manifestations of neuropeptide dysregulation, and we must begin with the architect of stress corticotropin releasing factor, or CRF.
CRF is the most extensively mapped neuropeptide in biological psychiatry. It is the master conductor of the mammalian stress response, synthesized primarily in the paraventricular nucleus, the PVN, of the hypothalamus.
And from the PBN, it drives the classic HPA axis. It travels to the pituitary, triggers the release of ACTH into the blood, which travels to the adrenal glands, triggering the massive release of cortisol to initiate the systemic fight-or-flight metabolic cascade.
But if we limit our understanding of CRF to just the peripheral HPA axis, we miss its psychiatric significance entirely. CRF acts extra-hypothalamically as a direct neuromodulator in the brain.
Where specifically?
It is densely expressed in the locus coeruleus, where it acts as the whip driving the noradrenergic arousal system. It's also highly active in the extended amygdala, specifically the bed nucleus of the striatum terminalis, which processes fear, anxiety, and threat appraisal.
So CRF does not just prepare the physical body for stress, it forces the subjective mind into a state of fearful vigilance.
Exactly. And there is a fascinating tug-of-war happening at the receptor level. The current paradigm suggests the two primary receptors, CRF1 and CRF2, serve opposing functions.
Let's contrast them.
The CRF1 receptor is the alarm. When CRF binds here, it drives arousal, anxiety, and the physiological stress cascade. The CRF2 receptor, however, which also binds related peptides called urocortins, appears to act as the counter-regulatory system.
So it calms things down.
Right. Activation of CRF2 promotes anxiolysis, anxiety reduction, and helps return the neural circuits to baseline after the threat has passed.
Let's apply this to clinical depression. The textbook states that hyperactivity of the HPA axis is one of the most robust, consistently replicated findings in the history of biologic psychiatry. Let's look at a real-world clinical scenario to test our reasoning.
Okay.
A 35-year-old female patient with a documented history of severe childhood neglect presents to the clinic with treatment-resistant major depressive disorder. If a clinician were to perform neuroendocrine testing and administer an intravenous injection of synthetic CRF, what would they observe and why?
They would observe a blunted ACTH response from the pituitary gland.
Wait, if she is severely depressed and her stress system is hyperactive, shouldn't her pituitary produce a massive, exaggerated spike in ACTH when provoked? Why is the response blunted?
It seems paradoxical until you apply physiological logic, specifically the concept of allostatic load and receptor downregulation. Because of her severe early life trauma and chronic depression, her hypothalamus has been hypersecreting its own endogenous CRF for years.
So the system is exhausted.
Exactly. The pituitary gland has been subjected to a relentless, toxic bombardment of CRF. To protect itself from total exhaustion, the pituitary downregulates its surface CRF receptors. It literally pulls them back inside the cell membrane.
I see.
Therefore, when you inject a bolus of exogenous CRF during the clinical test, there simply aren't enough receptors left on the surface of the pituitary to catch the signal. The blunted ACTH response is the footprint of a system that has been battered into submission by chronic hypersecretion.
That is a profound clinical insight. The blunted response is proof of the chronic internal storm. And we can verify this by looking directly at the brain fluid, can't we?
Yes, we can. If you perform a lumbar puncture on a severely depressed patient, you frequently find significantly elevated concentrations of CRF in the cerebrospinal fluid. This elevation serves as a state marker.
Meaning it fluctuates with the illness.
Right. If the patient is successfully treated, whether through fluoxetine, cognitive behavioral therapy, or ECT, those CRF levels normalize. If the levels remain persistently high despite a perceived improvement in mood, it is a highly reliable predictor of imminent clinical relapse.
But let's return to the 35-year-old patient's history of childhood neglect, because this is where the textbook transitions from biology to tragedy. The dysregulation of the CRF system is not just a random genetic mutation, it is actively constructed by the environment.
The evidence here is incontrovertible. Severe early life psychosocial stress, specifically physical abuse, sexual abuse, or profound neglect during critical developmental windows, permanently alters the trajectory and set point of the neuropeptide stress system.
It essentially hardwires the HPA axis to be hyperreactive for the rest of their life.
Exactly. Epigenetic mechanisms literally methylate the DNA, altering how the CRF and glucocorticoid receptor genes are expressed. Individuals with this trauma history exhibit exaggerated HPA axis responses to even mild psychosocial stressors in adulthood, and they maintain those elevated baseline CRF levels.
And the downstream consequences are even more devastating, aren't they?
They are. Chronic, unremitting exposure to high levels of extra-hypothalamic CRF eventually induces a protective shutdown in the nucleus accumbens, which is the brain's primary dopamine reward center.
So the stress system becomes so loud, so toxic, that the brain actively turns off the joy system just to survive the noise?
Functionally, yes. The dopamine neurons become insensitive to stimulation. This mechanism directly drives profound anhedonia, the total inability to experience pleasure, which is the most debilitating, treatment-resistant symptom of trauma-related major depression. The trauma doesn't just cause pain, it destroys a biological capacity for joy.
That is incredibly heavy, but so important to understand. So CRF shows us how the brain physically encodes isolation, threat, and despair. But how does the brain encode safety, connection, and love? This brings us to oxytocin, vasopressin, and the social brain.
Right. These two neuropeptides, meaning they're made of exactly 9 amino acids, are synthesized in the magnocellular neurons of the hypothalamus. And while we discussed their peripheral hormonal effects earlier, their extrahypothalamic projections into the forebrain, the amygdala and the striatum, are what govern complex, species-specific social behaviors.
And this brings us back to our hook, the voles. This research, spearheaded by scientists like Thomas Insel, completely revolutionized behavioral neuroscience. Walk us through the methodology and the implications of the prairie vole versus the montane vole.
So the prairie vole is a highly social creature native to the American Midwest. Upon mating, they form an enduring, lifelong pair bond. They are fiercely monogamous, the males aggressively defend the nest they co-parent, and they exhibit behaviors analogous to human empathy when their partner's stressed.
And the montane vole?
The montane vole, living in a different ecosystem, is physically nearly identical. But behaviorally, they are solitary. They do not form pair bonds. The male mates and immediately abandons the female, and they show no social preference for familiar partners.
If you look at the core mechanism driving this, it's not a difference in the volume of oxytocin or vasopressin in their blood, is it?
Not at all. Both species produce robust amounts of both peptides. The monumental discovery was that the difference lies entirely in the receptor distribution map within the brain.
How so?
In the monogamous prairie vole, there is an extraordinarily dense population of vasopressin V1A receptors located in the ventral pallidum, and dense oxytocin receptors in the striatum and nucleus accumbens. These brain regions are the core hubs of the mesolimbic dopamine reward pathway.
So the evolutionary brilliance here is that social bonding is circuitry.
Exactly. When a prairie vole mates, the resulting surge of oxytocin and vasopressin floods those specific receptors in the reward center. It triggers a massive dopamine release that structurally links the sensory memory of that specific partner, their scent, their touch, with the sensation of profound physiological reward.
And the montane voles just don't have that wiring.
Right. The non-monogamous montane voles simply lack those receptors in the reward hubs. The neuropeptides are released during mating, but the reward circuit is deaf to the signal. No dopamine is released in response to the social cue, so no bond is formed.
But the scientists didn't just stop at observing this difference. They wanted to prove causality. They actually manipulated the genetics.
They did. If we connect this to the bigger picture, what dictates where those receptors are placed in the brain during development? It is the promoter sequence of the receptor gene. The promoter acts as the genetic manager, instructing the cell where and when to express the gene.
No.
The prairie vole and the montane vole have distinct differences in their V1A receptor promoter sequences. To test this, researchers took the specific V1A receptor gene from a prairie vole, along with its unique promoter sequence, packed it into a viral vector, and inserted it into the brain of a non-monogamous mouse.
They genetically engineered a loner to possess the brain map of a lover.
And the results were staggering. The transgenic mouse developed a V1A receptor map identical to the prairie vole. And when placed in a social paradigm, this previously solitary, promiscuous mouse began exhibiting intense prairie vole-like social bonding and partner preference behaviors.
That is awe-inspiring, a genetic switch that fundamentally alters the capacity for attachment. So how does this translate from rodent models to human clinical manifestations?
Well, in humans, the system is immensely more complex, heavily modulated by the prefrontal cortex, but the core principles hold. In humans, oxytocin acts to enhance the signal-to-noise ratio of social cues.
So it makes social cues more obvious.
Yes, it amplifies the salience of interpersonal data eye gaze, subtle shifts in facial expressions, vocal intonations. Brain imaging studies demonstrate that when healthy volunteers are given oxytocin via an intranasal spray, which allows a fraction of the peptide to bypass the blood-brain barrier via the olfactory and trigeminal nerves, we see a marked reduction in amygdala activation in response to fearful or threatening faces.
Wow, so it directly targets the fear center.
Exactly. Oxytocin actively dampens social fear and promotes interpersonal trust.
Which immediately leads clinicians to consider conditions where social cognition is fundamentally impaired. The obvious target is autism spectrum disorder.
Right. Autism spectrum disorders, or ASD, are characterized by profound deficits in social communication, reciprocal interaction, and an inability to intuit the emotional states of others. Given the neuropeptide data, this system became a primary target for investigation.
Have they found genetic links?
Yes. Genetic association studies have repeatedly linked polymorphisms, variations, in the human vasopressin V1A receptor promoter gene to the diagnosis of ASD. Furthermore, there is emerging, though complex, evidence that children with autism may have altered concentrations of vasopressin in their cerebrospinal fluid.
So if the social filtering system is underactive or miswired, can we therapeutically boost it? What happened when they actually tried the intranasal sprays in clinical populations?
This is a highly active yet frustrating frontier of translational research. Clinical trials administering intranasal oxytocin and vasopressin to patients with ASD have yielded mixed but tantalizing results. Some studies show significant measurable improvements in social reciprocity, emotion recognition, and enhanced fMR activation in the brain's reward networks during social tasks.
But it's not a cure. What is the limiting factor?
Delivery and chronicity. The blood-brain barrier remains a formidable obstacle. Even with intranasal delivery, only a minuscule fraction of the active peptide reaches the deep cortical circuits. Furthermore, the brain adapts. Chronic administration of exogenous oxytocin can lead to receptor downregulation, potentially worsening the social deficits over time. We are tantalizingly close to understanding the biological roots of human connection, but pharmacologically, we are still using very blunt instruments.
We've seen how oxytocin filters social cues, bringing the vital signals of connection to the forefront. But the brain must execute another, even more fundamental kind of filtering raw sensory data. If that filter breaks down, we enter the realm of psychosis. This brings us to neurotensin and the biology of schizophrenia.
Neurotensin, or NT, is a 13-amino acid peptide that is intimately anatomically interwoven with the brain's dopamine system. In the midbrain, specifically within the ventral tegmental area, or VTA, neurotensin is synthesized and colocalized inside the exact same neurons that produce dopamine.
And where do those neurons go?
These VTA neurons project massively into the prefrontal cortex and the limbic system, the precise neuroanatomical pathways heavily implicated in the positive and negative symptoms of schizophrenia.
But it's highly specific. The textbook points out that in the neighboring substantia nigra, the area that controls motor movement and is destroyed in Parkinson's disease, the dopamine neurons do not co-express neurotensin.
That anatomical specificity is crucial. It means neurotensin is a modulator of cognitive and emotional dopamine, not motor dopamine. The prevailing hypothesis is that neurotensin acts as an endogenous, built-in antipsychotic.
An endogenous antipsychotic. That's fascinating.
It is. When researchers administer neurotensin to animal models, it produces behavioral effects that are virtually identical to administering a pharmaceutical antipsychotic drug like haloperidol. It suppresses hyperlocomotor activity, and most importantly, it dramatically enhances sensorimotor gating.
Let's unpack sensorimotor gating for the listener, because it is central to the schizophrenic experience.
Right. Sensorimotor gating is the neurological filter that allows you to maintain sanity in a chaotic world. If you are sitting in a bustling coffee shop, your brain is being bombarded by the sound of the espresso machine, the visual movement of people walking by, the smell of the pastries, and the conversation of the person sitting across from you.
That's a lot of data.
A lot of data. But a healthy brain automatically gates or filters out the irrelevant background noise, allowing you to focus your cognitive resources entirely on the conversation.
It dampens the sensory noise so the signal can get through.
Exactly. Patients with schizophrenia often suffer from profound, debilitating deficits in sensorimotor gating. They cannot filter the noise. They are relentlessly flooded by raw, unfiltered sensory data. The espresso machine sounds as loud and important as their own thoughts.
That sounds absolutely terrifying.
It is. This sensory flooding contributes directly to the cognitive fragmentation, paranoia, and psychotic delusions characteristic of the disease. Neurotensin's role in the VTA is to modulate dopamine to maintain that crucial sensory filter.
Let's put this into a clinical reasoning framework. A schizophrenic patient in acute psychosis is brought to the emergency department and is administered a standard typical antipsychotic, which works by aggressively blocking dopamine D2 receptors. What happens to the neurotensin system in their brain in response to this drug?
We observe a massive, sustained upregulation in neurotensin synthesis and release in those specific dopamine terminal regions in the forebrain.
The brain reacts to the dopamine blockade by pumping out its own endogenous antipsychotic peptide.
Precisely. It appears that a significant portion of the therapeutic efficacy of our standard antipsychotic medications may actually be mediated not just by blocking dopamine directly, but by triggering this compensatory surge in neurotensin.
Wow, so the drug forces the brain to heal itself in a way.
Yeah, and supporting this, several clinical studies have found that untreated first-episode schizophrenic patients frequently exhibit significantly decreased baseline levels of neurotensin in their cerebrospinal fluid. When their psychosis is treated and resolves, those CSF neurotensin levels normalize.
That makes perfect sense. Okay, so if neurotensin is trying to maintain cognitive order and filter the noise, what happens when a peptide's entire evolutionary job is to bypass the filters and flood the system with an unignorable alarm? Let's transition to a thematic grouping we can call the peptides of panic and pain. We'll start with cholecystokinin, or CCK.
CCK is an archetypal example of the dual-function evolutionary quirk. It was originally discovered in the gastrointestinal tract, where it orchestrates digestion by stimulating gallbladder contraction and pancreatic enzyme release.
But what does it do in the brain?
In the central nervous system, particularly in the cortex and amygdala, CCK is heavily implicated in the genesis of profound anxiety and panic.
And the human trials with CCK are genuinely terrifying.
They are. When researchers administer intravenous infusions of synthetic CCK fragments, or CCK agonists like penagastrin, it reliably and rapidly induces severe physical panic attacks in healthy volunteers.
It just immediately triggers a panic attack.
Immediately.
Yeah.
And in patients with a pre-existing diagnosis of panic disorder, it triggers massive panic attacks at much lower doses. It causes tachycardia, hyperventilation, a crushing sense of doom, and an overwhelming urge to flee. It is a pure panic generator.
Did they try to make a drug out of this?
They did. Consequently, pharmaceutical companies pursued CCK antagonists as novel anxiolytics. But unfortunately, they failed to demonstrate sufficient efficacy in clinical trials, likely due to bioavailability issues and the sheer complexity of the anxiety network.
Which brings us from emotional pain and panic to literal physical pain. Substance P.
Substance P is an 11 amino acid peptide that serves as the primary neurotransmitter conveying nociceptive painful signals from the peripheral nerves up the spinal cord and into the brain. But beyond acute physical pain, it acts centrally to modulate emotional pain. We see markedly elevated levels of substance P in the cerebrospinal fluid of patients suffering from severe depression and PTSD.
And this finding launched one of the most expensive pharmacological arms races in psychiatric history. The industry spent billions developing substance P receptor antagonists, specifically targeting the NK1 receptor, convinced they had found the holy grail, a novel, non-monoamine blockbuster antidepressant. What happened?
It was a spectacular failure of translational science. In animal models, the NK1 antagonists worked beautifully. They reduced distress vocalizations and behavioral despair. But in massive late-stage phase 3 clinical trials in humans, they completely failed to beat a placebo for the treatment of major depression.
Why the disconnect? Why did it cure the rats' depression but not the humans?
It likely comes down to the immense complexity of the human prefrontal cortex. Human depression is not simply the raw neurochemical perception of distress. It is intricately bound up in rumination, self-concept, and cognitive appraisal networks that are vastly more complex in humans than rodents and heavily modulated by serotonin and dopamine, not just substance P.
But the billions weren't entirely wasted, were they? There was a serendipitous pivot.
There was. While the NK1 antagonists failed for depression, researchers noted that the receptors were also heavily concentrated in the brainstem nuclei that control the emetic reflex, the vomiting center.
Ah, okay.
So they pivoted the trials, and those failed antidepressants became blockbuster drugs for the treatment of severe chemotherapy-induced nausea and vomiting. Drugs like aprepitant have fundamentally changed the quality of life for oncology patients. It is a true testament to the unpredictable nature of neuropeptide pharmacology.
Incredible. Let's round out this section by looking at the rapid-fire peptides that confer resilience. If CCK and substance P drive canic and pain, what systems actively push back? Neuropeptide Y and galanin.
Well, we discussed NPY earlier as the emergency brake on the noradrenergic stress response.
Yeah.
Clinically, a robust NPY response to trauma is highly protective. Military survival school studies have shown that soldiers who mount a massive NPY surge during extreme interrogation stress perform better cognitively and are vastly less likely to develop PTSD.
So low NPY is a risk factor.
Exactly. Low baseline NTY is a state of profound vulnerability. Galanin serves a parallel protective role. It acts as an inhibitory neuromodulator directly within the locus coeruleus, acting as a hyperpolarizing brake to prevent the noradrenergic neurons from firing themselves to death during extreme stress. Both peptides are vital molecules of resilience, actively shielding the brain from the toxicity of its own alarms.
And finally, a peptide that dictates our states of consciousness, orexin.
Orexin, also known as hypocretin, is synthesized by a tiny population of roughly 20,000 to 70,000 neurons located exclusively in the lateral hypothalamus. Its primary extrahypothalamic role is to aggressively promote and consolidate wakefulness across the cortex.
And the clinical manifestation here is one of the most direct causal links in all of neurology and psychiatry.
Yes. If a patient experiences an autoimmune event that selectively targets and destroys those specific 20,000 orexin-producing neurons, they lose the chemical scaffolding of wakefulness. This destruction leads directly to the sleep disorder narcolepsy, characterized by sudden, uncontrollable intrusions of REM sleep into waking life and cataplexy.
And we actually have drugs for this mechanism now, right?
We do. Armed with this highly specific mechanical knowledge, pharmacologists successfully developed orexin receptor antagonists. By specifically blocking the wakefulness signal, they created a highly effective, entirely new class of sleeping pills, the dual orexin receptor antagonists, like suvorexant, which are now FDA-approved and widely used for insomnia. They entirely bypass the GABAergic systems used by traditional drugs like Ambien or benzodiazepines.
That's a huge win. We've covered a dizzying array of molecules, circuits, and psychopathology. But as a clinician listening to this, the frustration must be palpable. If we know exactly how CRF drives trauma and how oxytocin drives connection, why aren't we curing these disorders? Let's delve into the integration and pharmacological frontier. We have to confront the shadow hanging over this entire field, the blood-brain barrier.
The blood-brain barrier is the evolutionary fortress protecting the central nervous system from circulating toxins and pathogens. It is composed of tightly packed endothelial cells that are highly lipophilic, meaning they are fat-based.
And neuropeptides don't mix well with fat.
Right. Neuropeptides are large, bulky, and hydrophilic. They love water, but repel fat. When you inject a neuropeptide into the bloodstream, it simply bounces off the blood-brain barrier. It cannot penetrate. Furthermore, even if you bypass the barrier, the circulating peptidases would shred the peptide within minutes. You cannot give a patient an oral pill of oxytocin or neurotensin and expect it to reach the target circuits in the brain.
So the entire strategy of modern psychopharmacology had to change. You can't use the peptide itself.
Correct. The pharmaceutical strategy absolutely relies on developing synthetic, small molecule, non-peptide drugs. To be viable, these molecules must be under 500 Daltons in molecular weight. They must be lipophilic enough to dissolve through the fatty blood-brain barrier. They must be structurally distinct enough that peptidases do not recognize and destroy them.
That sounds incredibly difficult.
It is. And yet, despite all those structural differences, they must perfectly mimic the three-dimensional shape of the endogenous peptide at the specific receptor site to trigger the precise cascade. It is an astronomical biochemical challenge.
And the industry threw immense resources at this challenge in the mid-2000s. The textbook details what we can call the big three failures. It caused a massive retreat from neuropeptide research.
It was a devastating era for the field. 3 massive, heavily funded Phase 3 clinical trial programs all collapsed around the same time. There were the CRF1 receptor antagonists, designed to treat broad, generalized anxiety disorder and major depressive disorder. There were the NK1 antagonists for depression, which we discussed, and there were the MPY-Y5 receptor antagonists aimed at treating severe obesity by blocking the feeding circuits.
Every single one of them failed to beat a sugar pill in the final analysis.
Right.
But the textbook makes a crucial argument. It argues that the drugs didn't necessarily fail because the biology was flawed. They failed because the clinical reasoning behind the trial design was fundamentally flawed.
Exactly. Why did the CRF1 antagonists fail for anxiety and depression? Because the trials relied on DSM-5 diagnostic categories to select patients. The DSM diagnoses a patient with major depressive disorder based on a phenomenological checklist of symptoms, low mood, poor sleep, and hedonia. But biologically, that diagnostic bucket is incredibly heterogeneous.
Right, you could have 10 patients in a room all meeting the DSM criteria for severe depression, but their brains are depressed for 10 completely different biochemical reasons.
Precisely. Patient A might be depressed due to chronic inflammation. Patient B due to an isolated serotonergic deficit. Perhaps only patient C, representing maybe 15 or 20% of the total population, actually has a hyperactive, trauma-induced CRF system driving their symptoms.
And if you test the drug on everyone,
If you test a highly specific CRF blocking drug on a massive, broad population where 80% of the people do not have a CRF problem to begin with, the drug will inevitably fail to show statistical efficacy. The statistical noise drowns out the specific signal. The trial fails, the drug is abandoned, but the mechanism might still have been miraculous for patient C.
so why did the broad clinical trials for CRF1 antagonists in generalized anxiety disorder completely fail? Because they treated a symptom checklist instead of a biological mechanism. But there is a breakthrough example in the literature that proves this point, and it signals the future of psychiatry. Tell us about the CRF1 antagonist trial for women with PTSD.
This study is a landmark in precision psychiatry. A trial was conducted using a specific CRF1 antagonist, GSK 561679, in a population of women diagnosed with PTSD. When the researchers looked at the primary endpoint across all subjects, treating the DSM diagnosis as a monolith, the drug failed. It showed no overall benefit compared to placebo.
But they didn't stop there.
No, instead of abandoning the drug, the researchers conducted a secondary, highly specific, genetically informed analysis.
They looked for patient C.
They isolated a specific biological subpopulation within the trial, women who had a documented history of severe child abuse, who were homozygous for a specific genetic variant, the GG genotype of the CRF1 receptor gene.
So they combined an environmental trauma marker with a highly specific genetic vulnerability.
Yes. Furthermore, they cross-referenced this with epigenetic markers, specifically looking at the DNA methylation status of the NR3C1 glucocorticoid receptor gene and the FKBP5 stress regulation gene. When they isolated that precise, biologically enriched subpopulation, the women whose depression was mechanistically driven by trauma-induced CRF dysregulation, the CRF1 antagonist was highly effective at significantly reducing their PTSD symptoms.
That is a total paradigm shift. It means the era of the psychiatric blockbuster drug is over. We are never going to have a Prozac for everyone again.
Nor should we want one. The future of psychiatric treatment relies entirely on precision medicine. We must move beyond symptom checklists and match the highly specific small molecule modulator to the precise biological architecture of the patient in front of us, informed by their genotype, their epigenetic markers, and their trauma history.
Let's touch on one final piece of future pharmacology mentioned in the text-advanced drug design, because it's not just about turning a receptor completely on or completely off anymore with blunt agonists or antagonists.
Standard orthosteric drugs, drugs that bind to the exact same main keyhole as the natural peptide, are often too blunt. They flood the system, disrupting the natural rhythm of the brain. Scientists are now designing allosteric modulators. These are drugs that bind to a completely different secondary site on the receptor, the allosteric site.
I think of it like a dimmer switch rather than an on-off switch.
A perfect analogy. Positive allosteric modulators, or PANMs, do not trigger the receptor on their own. If a PAM is sitting on the receptor, nothing happens. But when the brain's natural endogenous neuropeptide is released and binds to the main site, the PAM acts as an amplifier. It simply turns up the volume of the natural signal.
And negative allosteric modulator.
Negative allosteric modulators are names turn the volume down. This is vastly superior pharmacologically because it preserves the natural physiological timing and spatial release of the endogenous peptide. It only boosts the signal where and when the brain is already actively trying to send it.
And the text also introduces the cutting-edge concept of bias signaling.
Recall that a single G protein-coupled receptor can trigger multiple different intracellular cascades. What if one cascade is therapeutic, but the other causes terrible side effects? Biased signaling drugs are engineered to bind to the receptor and force it into a highly specific, unnatural conformation that only recruits one specific pathway, completely ignoring the others.
And the opioid crisis is the primary target for this, isn't it?
It is the holy grail of pain research. The mu-opioid receptor triggers a G protein cascade that provides profound pain relief. But it also recruits a different protein called beta-arrestin. The beta-arrestin pathway is theoretically the one that drives respiratory depression, profound tolerance, and the cellular adaptations leading to addiction.
So a biased ligand would.
A biased opioid ligand would be designed to trigger the G protein cascade for pain relief, but structurally fail to recruit beta-arrestin. In theory, it would be a powerful, non-addictive painkiller that cannot cause a fatal overdose. It represents the absolute pinnacle of rational drug design.
We have covered an immense amount of ground today, from the factory floor of the endoplasmic reticulum to the tragedy of childhood trauma, and finally to the genetic future of precision medicine. Let's distill the key takeaways from this chapter, establishing the core mental models for the clinical listener.
I would synthesize the material into 5 core principles. Number 1, neuropeptides are slow modulatory signaling molecules. They alter the resting tone of entire neural systems via diffuse volume transmission rather than engaging in fast point-to-point electrical signaling.
Right.
Number 2, they serve as the bridge between biology and meaning. They link physiological states directly to subjective emotional and relational experiences, encoding the psychological significance of a signal.
Number 3, corticotropin releasing factor, CRF, is the primary architect of the stress response. It is a system that is fundamentally, epigenetically, and permanently altered by severe early life trauma, leading to receptor downregulation and profound anhedonia.
Exactly. Number 4, oxytocin and vasopressin govern the social brain. Driven by precise genetic promoter sequences, they dictate attachment, bonding, and trust, representing the most promising, albeit difficult, targets for treating the social deficits in autism spectrum disorders.
And number 5?
Number 5, the future of psychiatric psychopharmacology relies entirely on precision medicine. The era of broad, heterogeneous clinical trials based on DSM symptom checklists is ending. The future belongs to matching advanced small molecule neuropeptide modulators, such as allosteric modulators or biased ligands, to specific patient genotypes, epigenetic profiles, and environmental trauma histories.
Beautifully synthesized. All right, let's test your clinical reasoning with a few single best answer questions. I'll read the question and options. We'll pause for a moment, and then the expert will break down the answer. Question 1. Which of the following best describes the primary mechanism by which neuropeptides differ from classical neurotransmitters like glutamate? Is it A, they are synthesized exclusively in the synaptic terminal, B, they are cleared from the synapse by highly specific presynaptic reuptake pumps, C, they generally act as neuromodulators via G protein-coupled receptors rather than directly opening ion channels, or D, they are always composed of more than 90 amino acids?
The answer is C. They are modulators using GPCRs. A is wrong because they are synthesized in the soma at the rough endoplasmic reticulum. B is wrong because they are degraded by peptidases. There is no reuptake. And D is wrong because they are 2 to 40 amino acids long. Anything over 90 is a protein.
Question 2. In the biosynthesis of neuropeptides, what is the specific role of prohormone convertases? A, transcribing mRNA from DNA in the nucleus, B, cleaving the signal peptide at the rough endoplasmic reticulum, C, cleaving the prohormone at specific basic amino acid pairs within the Golgi and secretory granules to yield the active peptide, or D, degrading the active peptide in the synaptic cleft?
The answer is C. Just think back to the factory line. The convertases act as molecular laser cutters in the Golgi and granules to cut the prohormone into active pieces.
Question 3. A 35-year-old female with a history of severe childhood neglect presents with treatment-resistant major depressive disorder. Neuroendocrine testing reveals a blunted ACTH response to an intravenous CRF challenge. What is the most likely pathophysiological mechanism underlying this specific finding? A. Primary pituitary failure to synthesize ACTH. B. Downregulation of pituitary CRF receptors secondary to chronic hypothalamic CRF hypersecretion. C. An overactive CRF2 receptor system suppressing the HPA axis. Or D. Reduced CRF concentrations in the cerebrospinal fluid.
The answer is B. Early trauma leads to chronic CRF hypersecretion, which subsequently downregulates the pituitary receptors, causing the blunted ACTH response to an exogenous challenge.
Question 4. Research on prairie voles versus montane voles demonstrates that species-specific monogamous attachment behaviors are most strongly associated with which of the following? A. Total volume of oxytocin produced in the peripheral bloodstream. B. The differential anatomical distribution of vasopressin V1A and oxytocin receptors in mesolimbic reward pathways. C. The absence of the vasopressin V2 receptor in montane voles. Or D. The inability of montane voles to synthesize neuropeptide Y.
The answer is B. Magic is in the promoter region genetics that dictate where the receptors are placed, specifically linking social cues to dopamine reward in the ventral pallidum and striatum.
Question 5. Which neuropeptide is highly colocalized with dopamine in the ventral tegmental area, functions to enhance sensoremogeting, and is markedly upregulated in target regions following the administration of D2-blocking antipsychotic medications? A. Cholecystokinin. B. Neurotensin. C. Substance P. Or D. Galanin.
The answer is B. Neurotensin acts as an endogenous antipsychotic-like molecule that helps filter sensory noise and is heavily modulated by traditional schizophrenia treatments.
Question 6. Why have broad clinical trials for CRF1 receptor antagonists largely failed to show efficacy in generalized anxiety disorder, despite robust preclinical evidence implicating CRF in stress? A. The drugs completely fail to cross the blood-brain barrier. B. The CRF1 receptor is not expressed in the human brain. C. GAD is highly biologically heterogeneous, and these drugs likely only benefit a precise subset of patients. Or D. The drugs inadvertently activated the CRF2 receptor, causing severe panic.
The answer is C. This highlights the future of precision medicine and psychiatry. The failure was in trial design and patient selection, relying on a heterogeneous DSM diagnosis, not necessarily a failure of the mechanism itself.
Excellent. And as we conclude, I want to leave you, the listener, with a final provocative thought. It's something to mull over on your own that builds on everything we've discussed today regarding the biology of meaning. We saw with the voles that a tiny microscopic shift in the promoter sequence of a single gene dictates whether that animal is capable of forming lifelong bonds or is destined to abandon its mate. We've seen how early life trauma permanently alters the epigenetic methylation of the CRF system, physically turning off the joy centers of the brain.
It really makes you think.
It does. If our deepest, most profound feelings of romantic attachment, our resilience to unimaginable stress, and our ability to look another human in the eye and feel empathy are heavily dictated by the spatial distribution and density of neuropeptide receptors, how much of what we consider our immutable personality, our moral character, or even our soul is actually just a biological reflection of our neuropeptide promoter genes?
It is a profound philosophical question wrapped entirely in a neurobiological reality. It completely challenges the very concepts of free will, personal responsibility, and identity.
It really does. I strongly direct all our listeners back to Chapter 1.8 of Kaplan and Sadok's comprehensive textbook of psychiatry. Review the tables detailing the amino acid sequences, look closely at the precise gene structures, and study the detailed neuroanatomy maps. It will solidify everything we've discussed today. Here are the details of this episode.
Neuropeptides, biology, regulation, and role in neuropsychiatric disorders. Byline: the slow signals of significance how the brain encodes stress, attachment, and enduring states.